I am Lauren Summers. I'm Senior Director of Lifelong Learning and Travel at the Yale Alumni Association. Thank you for joining this installment of our Fall Climate Change Conversations series. This is our second year doing it. And this year we have partnered with our colleagues at Harvard Alumni Travel. And we're very pleased to feature a number of speakers who have connections to both Yale and Harvard, as well as distinguished faculty from both universities. Together, we have organized 9 discussions for our collective alumni communities, each featuring experts across a wide range of topics relevant to climate change. All the lectures are being recorded and are available to view after the lecture is over, along with sometimes extra materials that our speakers provide. You can find that on the Yale Alumni Academy and Harvard Alumni Travel websites. Our next conversation will be on Monday, November 15th from 4 to 5:30 p.m. That will be our last conversation in this fall series and will feature Dean Sten Vermund from the Yale School of Public Health. He will be speaking on mitigating the public health effects of climate change. So we're closing out our climate change conversation series here with 2 conversations looking from the medical and the health point of view. Today, we're talking about climate change, the effects of climate change on virus emergence with Professor Paul Turner from the Yale School of Medicine. Dr. Turner is the Rachel Carson Professor of Ecology and Evolutionary Biology at Yale. He's also a microbiology faculty member at Yale School of Medicine. He earned his BA in biology from the University of Rochester and a PhD in microbial evolution from Michigan State University. He completed his postdoctoral work at the National Institutes of Health, the University of Valencia in Spain, and University of Maryland College Park, before he then joined Yale in 2001. Dr. Turner studies the evolutionary genetics of viruses, particularly phages that infect bacterial pathogens and RNA viruses transmitted by mosquitoes. He researches the use of phages to treat antibiotic-resistant bacterial diseases. He's very active in science communication outreach to the general public, and he's involved in programs where faculty collaborate with K-12 teachers to improve STEM education in underserved public schools. Dr. Turner also serves on the National Science Foundation's Bioadvisory Committee, and he is president-elect of the International Society for Evolution, Medicine, and Public Health. He is a fellow in the National Academy of Science, the American Academy of Arts and Sciences, and the American Academy of Microbiology. And in his extensive free time, he does lectures like this one for Yale alumni, right, Dr. Turner?
That was a wonderful introduction. Thank you so much. And thank you to all these very many participants who joined us today. This is fantastic. I mean, I guess I didn't know what to expect, but it's a delight that so many of you are with us. So I will begin by sharing my screen. All right, so this is my first slide, and just a reminder in this very colorful image of basically you are living very much in a virus world. And some of the threat of that is that virus diseases can emerge in humans. So the topic today is how might this be affected by a changing world, especially under climate change. So that's the topic. I'll mostly talk about this in a kind of an overview fashion. This is really a leading edge topic. It's something that's quite new, and therefore we have abundant more information to learn. I'll go through some more technical slides of the way that my group approaches this question, but the idea is certainly not to lose any of you in that presentation. I'd like to have everybody stay with us throughout and remain engaged, and look forward to questions and conversation at the end. So biodiversity, I'm an evolutionary biologist, and a lot of the reasons that I even entered my field is the appreciation for the world around me and how biodiverse it is. And I think that this is at least the reason that a lot of people go into biology as a discipline. So we know that in a changing world, unfortunately, humans are negatively impacting speciation and species on this planet and driving biodiversity down over time. So before we get to climate change, Let's think about, why does biodiversity matter in the first place? And there are very many reasons that I could put forward. Here are a few that are, you often see them in the literature. Just the productivity of the ecosystems which have diverse communities of species in them could be altered and diminished if some species are lost through time. There is an aesthetic reason to preserve biodiversity, is that it makes people feel good sometimes when they are in a place that is biodiverse and they see lots of species around them. There's something about that, doing the recreation and just appreciating the natural beauty around us. Survival of interacting species is threatened if any one of those species goes extinct. So you can have collapses in interactions because one species is lost. The disposal of natural wastes on the planet, so these are natural processes, these could be diminished as species are lost from ecosystems. And there are lots of economic benefits to having thriving, healthy ecosystems. This is often called ecosystem services. Now, the last is something that directly touches my research program, and is that through time, humans have made all sorts of amazing discoveries when they understand the natural world around them, including discoveries that improve human health. So in my case, I study viruses for very many reasons. Today, we're going to talk about dangerous viruses that cause disease, but some of the viruses that I study target only bacteria, and they are quite good at killing bacteria when antibiotics fail. So there's something that my group does, is that we tap the natural biodiversity of certain viruses on this planet. So global warming, I want to start here before moving on to climate change. A reminder that the long-term heating of the Earth's climate system that has been observed since pre-industrial period times, this is due to human activities and primarily fossil fuel burning that increases greenhouse gas levels in the Earth's atmosphere. So these types of sobering charts and graphs like you see in the middle of this slide, it's a reminder that this is something that is happening on the planet through our own activities. We're living in a warmer and warmer world on average, writ large across the globe. And the change in the global surface temperature relative to average temperatures prior to then are shown here. Now, getting to climate change itself, this is the long-term change in the average weather patterns that define local, regional, and global climates. So the primary drivers here are human activities, especially the global warming that I just mentioned. But this is also due to natural processes. So there are certain patterns through time on this planet, and events like El Nino, which I'm sure you've heard of, that these cyclical ocean patterns can also impact climate and also lead to changing climates. But we're focused today on human activities that are altering climate on this planet and climate change caused by those drivers. And the key indicators here that this is happening are that global land and ocean temperatures are increasing, and that sea levels are rising and ice is disappearing at the poles, and the frequency and severity of certain changes in hurricanes, wildfires, heat waves, and floods, et cetera. These do happen, but it's the frequency and the severity of the changes in them that are quite alarming. So climate drives redistribution of biodiversity. This is something that is talked about a lot, and it's a different problem than invasive species. Most people are aware of the fact that species can move around on the planet, including through human activities. But the problem of climate change is quite large, and it really affects the global ecosystem in which we thrive. So this process is influencing a greater proportion of Earth's biota through time than something like invasive species would. And the species range distributions are really what's at play here. So as organisms in their natural environments occupy a certain amount of territory, whether it's on terrestrial lands or in aquatic systems like the ocean, this is going to depend on how tolerant they are of the environmental factors and features that they encounter, and how well are they capable of moving to someplace else when environments become uninhabitable for them. So can they disperse or not? But all of that is happening in the backdrop of interactions with other species. And soon we will get to the interactions of humans with disease-causing species, pathogens. But let's just think about how climate drivers and changes in species distributions. I'll give you a couple of quick examples that probably you've heard of before, but if not, perhaps this will be helpful. So fish species may be driven deeper as sea surface temperatures rise. And that means that they could better elude some predators, or they would come into better contact with predators and face higher predation rates. But certainly in terms of fisheries and management, This is something that is worrisome because if you have species distributions of fishes moving about in the ocean in places that are harder and harder for fisher people to get to them, then you have a big problem. You can consider mountainsides. So as you think of a warming Earth and climate change affecting portions of the Earth and our organisms having to move up mountain slopes in order to find cooler environments that they're used to because their environments are changing and becoming too warm. So they could go upslope to escape warming lowlands. But as you can see in this image, the problem is that process can drive some species literally off the top of the mountain, and they won't have any land left to occupy because as species are moving up that hillside and that mountainside, those at the top could be entirely displaced. Now, ultimately, the real sobering thing is that as these changes continue to occur, because they're already happening, is it already too late? Are there some species that are so-called fated to extinction? And here, sadly, often a poster child is the polar bear. So these are large carnivores. They need to move about very large territory to find prey, and they can swim in open water, but they're not good at doing that for very big distances. And as ice flows melt and become less numerous in their environments, then there are not enough locations for these bears to occupy surfaces and to be on surfaces in between swimming events, and they simply cannot thrive to move about their environment to find prey items, and perhaps they'll be fated to extinction under climate change. But it is important to say that all of the predictions of climate change, some of them seem to be very lockstep with things that we're already observing, and others try to make predictions that are further off in the future. And of course, there's going to be some degree of uncertainty about exactly what is going to happen in the future. But we already think that there should be so-called winners and losers. And that's something that I think is important enough that we need to say it aloud. I've talked about biodiversity and biodiversity loss, but technically some human activities that are causing climate change and therefore threatening species with increasing rates of extinction, it is important to say that sometimes things would improve for humans along the way because species might be improving. So you consider the skipjack tuna in some Pacific Island nations. They are not, until recently, thriving near enough to those nations for those people to be fishing them in increased abundance. And if that is happening under climate change, one could say that that's a winning solution that is a good outcome of a very bad thing. However, I'll follow the good news with bad news. And timber production in places where people are used to thriving monetarily and economies are driven through timber production, if you have enough climate change altering distributions of tree species, then it's going to cause them to be unable to grow in some environments. And in places like Norway that are suffering declines in timber production already, this is likely going to only get worse and worse. So temperature and precipitation are important factors of climate. And we already know that these are important environmental drivers of infectious diseases. So now let's start talking about diseases as I move into discussing pathogen emergence. We've known for a very long time that waterborne diseases, if you have a lot of water in the environment, especially if it is not clean, and if humans are shedding disease agents like bacteria that can then go on and infect another person because they have to rely on unclean drinking water, then in circumstances like cholera disease, this is a very big deal. Now on the right, vector-transmitted infections have been a long plague of humanity. These are arthropods such as mosquitoes that would transmit them. And very famously, this has occurred for malaria, protist parasites. So as you have more and more changes in precipitation events that lead to better and better ability for mosquitoes to say have access to water, which they need to lay their eggs and for their larvae to develop, you're going to get higher rates of some arthropod-borne and vector-transmitted infections in certain places. Three other quick examples, beginning on the left, parasitic bacteria and helminths. It's a possibility that in places that are getting warmer through time and large mammals such as caribou and muskoxen and say the Arctic, it's already seen that they're suffering increasing rates of infection because there aren't enough cold periods, very cold periods for certain disease agents to die back. And instead they're doing constant infections in these creatures. And this is unfortunately a carcass of a muskox that died from this problem. And on the right, we've got marine and aquaculture diseases. These are a bigger and bigger thing through time and harder to keep those under control and obviously threatening the food supply. And the question is whether climate change in the future would make that worse. We certainly think that climate change, it does contribute to some ability of fungi, especially in sort of more humid environments. If you have changes locally that lead to better conditions for fungi to thrive outside of their hosts, then it's also easier for them to cause infections at higher rates in those places. There are lots of suggested drivers that are leading to amphibian declines by chytrid fungus. Too many for me to talk about today, but certainly climate change is something that is worrisome for fungal diseases such as chytrid fungi that are driving some amphibian species extinct. All right, so all of that is to say that climate change could alter disease dynamics, and it may potentially promote or exacerbate outbreaks in humans as well as in wildlife. Lots of scientific papers written as reviews in journal articles and also in primary literature, so-called, talk about climate change and its potential effects. I'm only mentioning a couple here that I turned to in producing this particular lecture that I found were pretty useful. But it's just a general way to now transition to talking about pathogens a little more in earnest here as we look at the problem of climate change and how it would impact the greater risk of emergence of diseases in the future that humans would suffer, especially virus diseases. So now in these reviews, there are several themes that are often mentioned, and I just should emphasize that something I said at the beginning is true. A lot of this research is currently underway. It's really at the leading edge, and therefore a lot of activity is going on. And you read these reviews and it says, we need more information. And that information can come through experimental studies on a wide variety of biological systems to try and get a handle on the problem as widely as possible across biodiversity. But determining whether diseases would shift, increase, or decrease with climate change, that's really the root of the problem. And there's a high degree of uncertainty for any particular disease agent, some species. How is climate change going to affect it? Last, you can take a theoretical or mathematical approach. So a lot of models attempt to describe things that are observed in nature. A lot of models attempt to predict into the future what will happen, but they don't always bring in so-called mechanisms. They don't always bring in, the real nuts and bolts of what biological systems have at their disposal to solve problems, say through evolution or through ecological interactions with other organisms. And ultimately, how is this shaping the traits that would evolve through time? So some of these reviews are calling for better models to understand the drivers of climate change and how they are interacting with disease systems to cause worse problems in the future. Now, a lot of this research is already focused on range expansion. So as you have a warming Earth, as you have some differing climate patterns, essentially, let's start first with arthropods. Arthropods are very important for transmitting diseases in a variety of host systems, including humans. So we've all heard of ticks and mosquitoes and other creatures that are arthropods that can transmit pathogens between humans. And the problem is, in our ever-changing world, the range or the geographic ability for these species to thrive is increasing. And therefore, necessarily, you have more and more humans at risk of the infectious diseases that these organisms could transmit. So in this lower right-hand image, it's talking about as CO2 is increasing in the atmosphere, the ultimate effects on global warming and climate change. It is already seen that there's a growing distribution of certain important mosquito species that are known to transmit very dangerous pathogens. And this is certainly going to put more and more humans at risk. In this particular example, it's right in my neighborhood, literally. So this is in the Northeast United States where we would be worried about in this particular projection map. Globally, though, there are plenty of places that are going to be affected by this problem. The range expansion of arthropod vectors that transmit infectious diseases is just generally expected to increase, and therefore, especially around the equator, as you expand into the latitudes away from there, then necessarily more and more people will be suffering with problems that we already know are big ones in certain parts of the world, and they're going to become new problems for public health and biomedicine in other parts of the world. All right, so the hallmarks of climate change are increased variability of temperatures, and precipitation, etc. That's what a lot of climate change models predict, is that environments become less predictable over time, more volatile. And so this produces a particularly big problem for biological species, because all the time, no matter what species you can think of, there is this interaction with the environment to improve in fitness if possible. But it could be that the environmental challenges can't be met through evolution, and therefore a species would go extinct. And the main question that I want to now continue to with my lecture and focus on for the remainder is really, are unpredictable or stochastic environmental changes that are expected to constrain the ability for species to adapt, are they going to be able to persist in the face of this? Or will you get many, many cases like this polar bear where the species seems to be perhaps fated for extinction? Now, as an evolutionary biologist who studies disease pathogens, I have to take that question and think about it in my system. So how would a virologist like me study this problem? The goal in predicting emergence is that we want to get more accurate with it. We want to know the potential for disease agents to shift onto new hosts. We want to know why some pathogens are successful at infecting new or multiple hosts. And we want to know the rules that govern virus evolution, adaptation, and constraint and extinction if we're very worried about virus pathogens emerging, as we're dealing with in the current pandemic. So this slide is showing some pretty obvious information when one considers recent coronavirus, emergence events in the current pandemic, HIV that shifted from non-human primates into humans, and influenza virus, which is a pathogen that we see all the time, but we're on this constant treadmill of trying to keep up with its genetic diversity to find vaccines and to develop vaccines that keep pace with the evolution of these viruses. And here I'm reminding with these images that the majority of the Earth's population of influenza viruses is not in humans. It resides in reservoir species for us, and that would be especially wild waterfowl. So the enduring concern is that we could have zoonotic events or the jumping of those influenza virus variants into humans, for which we have poor ability to control them with our own immunity, and perhaps very poor immunity to control them with vaccines. So RNA viruses are overrepresented as emergence problems. I think I can say that with some confidence. I study RNA viruses, and probably for many of you participating today, this is news that you're familiar with. So if you look at emerging and pandemic pathogens of humans in the recent times, as this chart is illustrating, if you go back to 2000 to the current day, there's something that's quite obvious in this when I point it out to you, is that 9 out of these 10 are RNA viruses. And it is not thought that RNA viruses make up nearly the majority of viruses on this planet. Most of viruses on this planet seem to have DNA genomes, but RNA viruses, because they have RNA genomes, they mutate more readily. RNA doesn't have error correction generally, so they change very quickly. And in this particular chart, it's talking about deaths from major epidemics on a log scale. So this is a lot of human lives lost. And only one of these is due to a non-virus pathogen, in this case, cholera bacteria. So this is really something that we need to be ever more prepared for in the future. So can RNA viruses adapt to the rapidly changing environments that are predicted by many climate change models? That's what I'm going to take you through this thought, and looks for some evidence of answers to this question from my own research. And let me preface it by saying some of this will be technical details shown in graphs, and I'll walk you through it. And the idea in the next 8 or 9 slides is certainly not for me to lose you, but I'm going to be purposefully kind of superficial at times in order not to lose you. I welcome your questions if you need details at the end. But the point is, I want to now show you some evidence of how a researcher like me would try to get a handle, at least in the systems that I work on, for some evidence that speaks to this question, because this is really an important question as we move forward. All right, so now how would you do this? Let's think hypothetically of some evolving lineage, and it's going to be a virus, and it's moving from the left to the right on your screen here. If you look at the top example, if a virus lineage has to see different environments, different temperatures through time, different hosts through time, necessarily it might be kind of generalized to deal with this fluctuating environment. If it sees only a constant environment, then it kind of has the luxury of only evolving to worry about that environment and become as fit as possible within it. So a lot of my research, as shown here on the lower left, I'm just adding a numerous A lot of citations there for anybody who's interested, but there's lots of ways to study this. I and others have looked at in the laboratory, can we alter environments to keep them constant or to be fluctuating through time, and look at RNA viruses, such as the ones pictured here on the lower right. How do they deal with these problems? And generally what we find is that they have no problem at all evolving in alternating environments, that they're quite good at their speed of evolution, their ability to rapidly evolve to deal with this. So they do have a lot of the evolutionary potential for which their overrepresentation in emergence and pandemics is justifying, that these viruses are very good at being the champions of evolution. Sometimes people call them this. But here's a distinct challenge that is very seldom studied, and we sought to look at it because this is what some climate change models are predicting, that if you take lineages of some systems, including RNA viruses, and you try to challenge them with unpredictable environments, as you see in this lower depiction here, then what will they do? Can they still adapt when faced with environmental uncertainty? And if so, that would be rather sobering news, because we would think that they would be maybe better capable of causing emergence and pandemic problems in the future, because they are able to deal with this problem, and maybe host organisms like us would not be able to deal with it. And it kind of further tips the advantage to RNA viruses and the evolutionary interactions with their hosts. So how would one do this? I'm going to now walk you through a study or two. I think I have two to quickly present. And largely we'll do it with model systems. And this is a virus, vesicular stomatitis virus, that is important in animal agriculture. It actually causes vector-borne disease in horses and cattle, etc. It's an RNA virus of mammals, but not an important one in humans. So it's safe for us to use it in the laboratory. It is vector transmitted, so just keep that in mind. And that image on the images on the left are to describe that. But on the right, I'm showing this cartoon image of this bullet-shaped virus particle, and I'm reminding myself to say it doesn't really have much genetics at its disposal. It has 5 genes. You know, that's not much. And yet this virus and other RNA viruses that have similarly small genomes are really good at adapting and really good at evolving. So let's take this virus into a challenge to see how would it deal with environmental uncertainty. And we can easily do this in the laboratory, because we can grow these viruses on host cells that are cultured and they're of mammalian origin or insect vector origin, and we can look at them in tissue culture in the laboratory. And one might design an experiment like this, which was done by a former postdoc of mine, Barry Alto, who's now a professor at the University of Florida. So typically we would do something called experimental evolution. We would take an ancestral form of this virus in the laboratory, and we would put it into an experiment like this one, where here on the left, if we were caring about the ability of lineages, and here are 5 of them shown in red, of this virus, and if we evolve it over time for 100 generations in a warm environment, that's a pretty typical human body temperature, 37 degrees C. We would like to know, how does that virus do? It's used to seeing mammalian body temperature, but let's see how much it could further improve. Whereas you could look at kind of the other edge of the niche space, so to speak, a very cool temperature, much cooler than body temperature of most mammals, and see whether this poses a particular challenge for this virus. But really, it's the 2 environments in the middle that we care about. The one in green is a typical alternating environment where we shift the virus lineages between those 2 extremes. But the random environment is the one that is going to be illustrating something about a changing, unpredictable environment. If we come into the laboratory, and this is what Barry did, and he's going to use what's called a random number generator, and he's going to set the temperature of the incubator to whatever that random number generator said for that day. And it's going to challenge those lineages in purple to keep pace with a very unpredictable environment through time. All right, so the way that we kind of measure the currency of the change, I'll just quickly walk you through this. Imagine now you've got this bullet-shaped evolved virus from his experiment, and we can also go back to the ancestral one that started the whole experiment and take it from the freezer. You can literally put them together on the same set of cells and enumerate at the beginning what's their relative ratio. And just to confirm that you put them together at a 50-50 ratio, for example. And then you let them incubate to allow virus growth. And after time, you see which one does better. Is there anything about testing the evolved virus in its own environment that it just evolved in to prove whether or not it improved in fitness relative to the ancestral form? And in the end, the ratio of these 2 ratios are something that we call fitness. It's sort of a dimensionless metric that tells you how well did this evolved virus do in some environment after the evolution took place. And now I'll quickly go to a set of results that we published back in 2013. Now, there's a lot going on here, but on this y-axis, this is how fit are these viruses at the end of the experiment at the high temperature of 37 degrees versus how fit are they at the lower temperature of 29 degrees. And the dashed lines are showing the ancestral values, and we set that to 0. So what you can see is here in the middle, those that were evolved strictly in a warm environment of red or a cool environment of blue, it's interesting that they lie above this dashed line and to the right of this dashed line, meaning that they've improved relative to the ancestor in both environments, even though they only evolved in one environment. So whatever performance they improved in that single environment dragged them upward in fitness at the other edge of the niche. Now, interestingly, if you take those virus lineages and put them in an alternating environment, they also improve relative to the ancestor, but you're sort of challenging them more because they're dealing with multiple environments and they're changing in different ways than the blue and red ones. And you can see that they're actually outpacing the rate of evolution in this 29 degree C environment and a little bit to the 37 degree environment because they're challenged to see both environments and improve at them at the same time. But here's where it gets interesting. These in purple are sitting right down here near these intersecting dashed lines. That means they really haven't improved relative to the ancestor. And it shows us that even though these champions of evolution, these RNA viruses, are very good at evolution, this kind of unpredictable environment is pretty challenging for them. And in this particular experiment, they don't really show any improvement when facing that challenge. All right. Now, what we do generally at the end of such experiments is catalog up all of the mutational changes that separated those lineages from the ancestor. Here across the top is the virus genome. And again, it has only 5 genes, which I'm abbreviating by single letters. It also has this untranslated region where sometimes a mutation can occur. So what you're seeing here is for the 29C evolved and 37C evolved, et cetera, virus lineages, and remember there were 5 per each treatment group, where are the mutations happening? So the nice thing in this experiment, it was quite satisfying that already in the literature, we knew that it's kind of the equivalent of the spike protein that you hear about a lot in SARS-CoV-2. This glycoprotein is the important protein that the virus uses to attach to cells. And it's already known in the literature that that can be responsible for temperature sensitivity. So it's not surprising to me, and I'm highlighting it here by circling G with this red oval, that this is where a lot of the action occurred mutationally. So here you see lots of mutations happening at that locus that separates these lineages from the ancestor. But one other thing I want to draw your attention to is this purple oval. So you may not be that impressed with it, but what it's summarizing is that on average through this genetic screen, you see more mutations happening on average for the viruses in this random treatment than you see in the other ones. So in other words, they're changing, they're evolving, they're just not improving in their environment. So let's look at that in a deeper way. And the way to do that is through what we call deep sequencing. You take the same viruses and you probe at a deeper level to find even the rare mutations that occurred through time. Again, this is a bit busy. Across the top, this is a heat map to tell you how deep do you have to go to find a mutational change, how rare is it in that population. Again, we've got the reference here of the virus genome, and the ancestor itself has some differences that separate it from what's called the canonical genome in GenBank. Don't worry about that. It's really all this other information here that is suggesting that you can use deep sequencing to look at a lot of these changes at a deeper level and catalog how many of them are occurring in these different populations. And what is it showing? It's actually showing that deep sequencing reveals that the random populations indeed have greater variation. They're not improving in fitness, but they've amassed greater variation than these other lineages. I'm highlighting that for the lineages that were evolved at the warm temperature and those that were evolved in these random temperatures. So now the next experiment, and that's where I'll get to the ending, is how much of that change might matter in a future world. Imagine one that got very, very warm. So is there anything about this experiment, even though these random evolved lineages do very poorly in this particular experiment, have they actually accumulated mutations that might help them in another context? So this was the work of another former postdoc, Andrea Gloria Soria. And she did an experiment that followed berries. So berries is at the top. I showed you that earlier. So now if we just key in on the lineages in red and in purple, you can sort of take all that genetic information, put it together into one gene pool, and create 5 new lineages from it. And you could put them in a very, very challenging environment for a shorter evolutionary period, but you could see how well would they do in a very, very warm world of, say, 40 degrees C for them, very warm world for them. And I'll say, without showing you the data, I'll just tell you straight up that we knew initially in this experiment that when you set this experiment up, that all these lineages do very poorly at 40 degrees C. So they have a lot of room for improvement. How did they improve? This is what we got as a striking and very unexpected result that we published last year. What we saw here circled in purple is that the height of these symbols on this graph say that they have improved to fitness at a particular level. And these are all these lineages that were prior evolved under random environments. And now you challenge them at this warmer environment. They've now outpaced the evolution of lineages that actually evolved in an environment not that different from 40C at 37C. And it's also showing you here the ancestors of these two groups and showing how poorly they were at the beginning. And actually those in red didn't improve very much. So what's going on here? Again, we turn to one of these very perhaps confusing charts, but it's just cataloging up the genetic changes. And I'm just going to jump to what's going on here. What we found is that, again, here's our bullet-shaped virus. And now I'm pointing to something that was unexpected. This is what's called the replicase. This is the protein that drives the whole replication of this virus when it is able to enter host cells. And those viruses that evolved at 37C, and then you challenge them to evolve at 40C, they seemed very constrained to only undergo certain further evolutionary changes and mutations that are happening in this L protein and a little bit in the glycoprotein. But here in purple, there's not much so-called parallelism occurring for these viruses. Instead, there's lots of mutations all over the place here relative to the lower group. And it says that there's something about the amassed variation genetically in those lineages that predispose them to use that variation in a new environment to a greater extent than organisms that were arguably, or I should say microbes that are arguably better adapted to them. And they've literally leapfrogged. So do stochastic or unpredictable environments constrain RNA virus adaptation? The first experiment would suggest yes. Environmental stochasticity or unpredictability is something that led to these maladapted virus populations, and maybe we could take some comfort in that. Is that good news that viruses will struggle with this problem as well? But the plot thickens because unfortunately, the poorly fit or maladapted viruses were the ones that evolutionarily leaped over and became better fit and better adapted than their counterparts that were kind of sitting closer to the niche edge. And that's bad news. It means that the times today that are grooming viruses for what challenges they may see in the future, it's perhaps very unpredictable what will happen next. So I'll end with saying a little bit about how can you bring greater realism to this? Well, we try to do this even in our own research by maybe doing field studies on, in this case, vector transmitted systems that have already changed maybe in a warming world and under climate change. And an example would be dengue virus, which is arguably the most important vector transmitted emerging virus on the planet. And a lot of projection maps are already showing that its range distribution, this mosquito, has increased, and therefore more and more people, such as in South America and increasingly in the US, are going to be exposed to dengue virus. So what we've done is go into what are considered warm parts of the world, like Vietnam, and we've taken mosquitoes from the north versus the south of Vietnam, and these dengue viruses from the north and south of Vietnam. And here on the lower right, we're finding that viruses from Hanoi and Ho Chi Minh City, in those two different extremes for temperatures that are seen in this country, they are already showing evidence of evolutionarily changing from one another and becoming what's called divergent. So they're keying in on local environments, presumably the temperature, and the mosquitoes that are reared at those temperatures as the best fit. And if you mix and match them around, they have some challenges. So I'm not really showing you the data from this paper, but you can go see it if you want. The point here is that you can do all this in the laboratory where you're bringing field caught mosquitoes and viruses from clinical samples in different locations and trying to see evidence of, is there already temperature adaptation? And can you make predictions in systems like these about what happens in the future? So I've come to the end of my presentation, and thank you if you hung in there with me. I hope that that was understandable. And I look forward to questions about that and conversation that follows. And I think I'd better stop sharing to facilitate that. And if you have anything that you'd like to ask me, I'd love to hear it.
Okay, thank you, Professor Turner. Super impressed with how complicated that presentation was, and yet how easy it was to follow and understand. So I know we've got some time here for Q&A. So I invite you, if you have any questions, to please post them in the Q&A box. Any question is fine and welcome. And I want to start with a question from Christina, who asked if your virus fitness work is the same as gain of function studies.
That's a hard answer. That's a hard question to answer. I mean, gain of function studies have understandably been problematic. So if you take a pathogen and put it into an experimental setting where you are, in the end, selecting for changes in that pathogen that make it more dangerous, then it's put into this context of gain of function, and those experiments are not allowed. What I just showed you are completely allowable experiments. It's basically taking a model system in a very confined laboratory where it's not capable of escaping. In fact, everything that I showed you, there were no, for the experimental part, there were no mosquitoes being used, right? We were just evolving the viruses to look at their potential. So technically, you're taking an arthropod-borne virus in that experiment and doing your best to get some insight, but you're never putting it in mosquitoes. So therefore, I would not call it a gain of function experiment. It's a good question and a difficult one sometimes to walk that line. And we always want to be ethically responsible, as well as, we don't want to put anybody at harm, including ourselves. So it is something that is important to keep in mind. So thank you for that question.
I want to go back to, I don't want to put you on the spot, but there was a slide that you showed that I want to go back to if we can. It was a slide that showed the number of mutations in the various viruses. And it,
you want me to show it, or?
Yeah, if you don't mind.
Sure.
It was showing the SNP mutations in the various viruses.
So I had various, oh, probably you're talking
that one.
Oh, this one? Yeah.
So what I found interesting about this slide was that if we look at these three, the red, green, and purple, we're seeing that, well, and the blue, right? We're seeing a large number of mutations, but what you were saying on this slide is that none of them really matter to.
Oh, well, some of it, like, imagine, so what you're seeing for the lines that are consistently across the entire chart, that indicates mutations that happen in this virus when you tend to grow it in the laboratory. Okay, so those mutations don't really have much to do with the particular treatments, and we can prove that, but our automatic inference is that they don't matter for the treatments. They matter for the fact that you're now growing the virus in the laboratory. So it is a tricky thing in virology to distinguish between the mutations that matter in an RNA virus system that readily evolves versus those that matter in the context of the environments that you're controlling, and you're making the viruses challenged to improve within them. It's kind of two things at the same time, and some of the information is useful for the main hypothesis, and frankly, the rest of it is kind of useless because it's not really your focus. Does that help?
Yes, but I think what I was getting at is this idea that the viruses are mutating in response to being challenged.
Oh, yes.
But mutating in ways that don't really directly affect their ability to adapt to climate change. But somewhere down the line, the fact that they have this wide variety of sort of useless mutations.
Good question, Lauren.
Seems to make them much, it's sort of like this Darwinian evolution, right? Like they evolve all these sort of useless mutations.
Right.
but they are now the fittest in adapting to the environment. It seems like that was one of the main points of your presentation.
Yes. So evolution uses whatever is at its disposal for natural selection to solve problems and have biological populations improve in their environment, if possible. So the currency to do that is there. And it's very hard to predict for any mutation versus some other mutation. If you put an organism into a new environment, would that mutation be sort of a driver of further adaptation? That's an enduring core question in evolutionary prediction, for which it's very difficult to refine that prediction. But what you said is absolutely correct, and part of the enduring problem of predicting emergence, because we can catalog easier and easier genetic variation in experiments like this, or frankly, from surveillance, right? You can go look at bats that are nesting in some cave, and if you sample those individuals, and if they all happen to have a similar virus, they're likely to have different variants of those viruses within them. But the question is, do we care about any one or more of those variants for the potential to emerge in another host, such as humans? So that's very, very hard to predict. Okay, and I have to say that's the sobering side of, you're going to see more and more surveillance work done in disease systems. And the question is, with that information, what can we really do with it? So I really liked your question. That was quite
well. And I like this slide because I think it shows this sort of great diversity that can evolve in the genome of the virus. And basically, it says in a very simple way, the best thing we could do is not stress the viruses out.
correct. Because they're going to mutate anyway, and they may not improve in fitness, but it's this accumulated variation that they have that it's very difficult for us to say, oh, is that variant just automatically disposed to better navigate the human immune system for some reason? And the problem is we have uncertainty for our existing virus pathogens, how they even navigate the immune system to successfully cause disease. So without knowing that, it's quite difficult to predict when you measure any one variant, even that same species. We're getting better and better because we have to. This has been a huge uptick in our ability to do this because of the research that's come about during the pandemic.
Well, and a follow-on question is, in one of your earlier slides, you talked about the range of, for example, mosquitoes, these vector arthropods, the range that they have expanding.
Mm-hmm.
And it would seem that if their range expands, that they somehow evolve to be more fill in the blank, more anything, which creates an opportunity for the viruses to evolve within them.
Perfect. Yes. So the range expansion of mosquitoes, such as here in the Northeast, where I think for the first time in Connecticut, we've seen in the wild some mosquitoes that are quite good at vector transmitting very dangerous pathogens that they've never been found in Connecticut before. Okay. And I could say the same thing at different points in time in this region and give you examples of that. So what it means is that now this ecosystem is predisposed that if dengue virus or something else comes in, it can be transmitted because the mosquito vector is already here. And the question is that really possible? That if these mosquitoes, they basically are worried about nighttime temperature. And if nighttime temperature gets too cold, they'll die because they can't control their body temperature. And in a global warming world, the problem is that the nighttime temperatures are creeping up. So if some virus pathogen comes in, as you asked about earlier, could we predict that variant A versus B versus C, if it comes in here, would it actually get established? Would it be able to thrive through all the other environmental details of the northeastern, northern Atlantic coast area of the United States? And that's where laboratory experiments can give us some information about the variety of variants of a worrisome pathogen, such as a virus, and looking at its ability to thrive under conditions that we know are representative of places like coastal Connecticut, et cetera, because it's kind of a waiting game. at this point for some of those pathogens to actually arrive here and be transmitted around, and it could happen successfully. So it's lockstep with what I said earlier about the geographic range expansion of arthropods for vector transmitted diseases is just simply not good news. It's a bigger biomedical problem looming.
Well, I know I was bitten by at least 5 or 6 different kinds of mosquitoes this summer. So
Me too.
I can give anecdotal support too.
There are plenty of mosquitoes, sadly, in our region.
Yes. There are new varieties of mosquitoes that probably were not here last summer even.
Yeah, this is true. Okay.
I want to go to Peter's question. Peter's question. How would you address host evolution, for example, in an arthropod vector in terms of assessing overall fitness or ineffectivity of a disease-causing virus? So it's a little bit of a follow-on of what we're talking about.
Sure. Yeah, this is where you could bring in a model host, like a rodent. So if you have in a system set up in a laboratory or some sort of a controlled setting where you have not only the pathogen and the mosquito, but you also have live mammals for blood feeding events. But the problem is it gets very large and cumbersome and expensive. Something that I spoke about this earlier today at a Yale event. I'm really excited about a potential kind of research that we might do is in collaboration with some folks at Johns Hopkins, where they have tried to establish artificial wet markets. So if you take organisms and you put them in cages, such as they would be found as organisms straight from the wild in a wet market, and if you have other species that are able to move around that environment freely, like a bird that could fly and could move pathogens around that are either directly transmitted, or you could also put an arthropod in there like a mosquito, then you essentially set up a system that is mimicking what we think is a troublesome situation. And you're putting live organisms in there that are the hosts. So through time, what we want to do, if we can get the resources to do it, is to scrutinize those sorts of systems through time to see how would a pathogen that comes in there emerge, and what are the evolutionary rules or the genetic changes that let it do this, and what are the so-called phenotypic or fitness trait differences that let this happen. What's going on in the hosts in terms of their immune system, and how are they changing in their lifetimes or not to deal successfully with these problems. And as the question asks, though, it really gets difficult if you want to look across generations of hosts, because in general, host generations like ours, they're much longer than the generations of the pathogens that are causing disease within us. So you have to be very crafty and get around those problems in nimble ways, but I'm just admitting that it's pretty challenging to do both the host evolution per se and the pathogen evolution alongside it, because it's just much easier in human time to do the pathogen evolution. So I hope that kind of answered the question. I'm not sure if it did.
I think it does. And this is a different Peter. Has I think what's a follow-on question. Beyond temperature for in vitro growth and replication, what other environmental variables that relate to current climate change issues can you test for in your VSV model system?
Yeah, and the VSV model system, at least with the kind of experimental setups that we have, they can be limited, but we could look at effects of humidity, for example, and also altering humidity. And then, you could do things that the problem with climate change is that multiple things are changing either at the same time or not at the same time. So you could look at both temperature and humidity, or humidity and temperature alone. So you can start to kind of play around with complexities of different things changing at the same time. It gets more challenging to maybe consider precipitation, which can, of course, contribute to ambient humidity. But I really like that question because it's a reminder for me to say, look, no experiment is going to mimic nature. I think people have given up on that a long time ago because it's virtually impossible. And it's really not the point of most empirical research, is we want to look at important factors of the natural world and try and key in on them to see if we can prove that those are the drivers, those are the factors that cause particular changes to happen that we're predicting. I just cannot create everything of the real world inside of a laboratory and this is the enduring problem as we study disease, is that just it's impossible to create that ecological complexity. And yet we make strides all the time, right? I mean, don't get me wrong on this. So we do the best we can. But questions like this are the right ones. I think that people are asking and considering in the very complex scenario that we're not quite sure how the pandemic ultimately started and the exact drivers of it, but we are for sure worried about it happening in the future. So we would like to get some realism to those experiments while admitting that we cannot get the reality of the world that where these events actually occur.
Okay, I want to just, Peter had a lot of questions, so I want
Okay.
to get Peter's question in, and then I'm going to go to, after that, Glenn had a follow on to a question I asked earlier. So Peter says, since viruses require infecting a host to replicate, is varying incubation temperature, as you've done in the VSV model, the most appropriate surrogate for environmental variability, since the host animal body temperature is unlikely to vary greatly over time?
Yeah, that's a good question. And I guess it speaks to, we're trying to look at the effects of the temperature itself on the viruses. And that is, how do viruses adapt under constant alternating or random temperatures like I showed? So I always try to be safe and not over claiming, right? Not over concluding from what we observe. So now we have evidence that if I manipulate temperature in these ways, in this particular RNA virus system, this is what happens. But it is, one of the things, as a virologist, the thing that concerns me is that often you hear people talking about viruses kind of in toto as the same thing, and that the same rules would apply to them, and no way. That's just not, that's like me saying, well, the same rules for a lion should apply to a zebra. And that's just not the case. One is a carnivore, one is an herbivore, and so on. So as we learn more about the amazing biodiversity of viruses on this planet, for which the vast majority have zero interaction with humans, but we do get insights into differing reproductive strategies of viruses within their hosts. How are they escaping or hiding from host immune systems and other defenses? So this is like a never-ending possibility, at least in the near future, to catalog the virus world and learn from it. And I just have to say that that should help us. We had SARS-1, we had MERS, if people know what that is. It's jumped from camels into humans and causes 30% case fatality rate. So those two coronaviruses are themselves quite different from SARS-CoV-2. And my point is, it's hard to say it as you work your way through a pandemic. It could be worse. Right? We now know of, say, MERS is actually, it would be a much worse circumstance in many ways. Of course, if you have 1 in 3 infected people likely dying. So the point is that as we do more and more work in virology, just to understand viruses and their complexities and the different ways that they've strategized through evolution on how to exploit hosts, we're necessarily learning more information that could be useful in the future if a virus like that is something that emerges. And sorry, not to belabor the point, but one more thing is the enduring problem of this pandemic is the combination of ease of transmission and high infection rates with almost no trust in the symptomology, right? Because if you're asymptomatic versus winding up in the emergency room, it's the same virus. And that was something that was very hard to predict. I mean, maybe there's some of those out there, including on this call, who said that they predicted it 15 years ago, but the vast majority of people who I know who work in infectious disease work, is that's a combination of traits that we hadn't really been on the lookout for. And my point is that as you survey the virus world and understand how viruses strategize to exploit their hosts, you're going to necessarily have more information that goes into our arsenal, so to speak, of understanding what viruses might do next.
Yeah, I'm glad that you actually belabored the point, because I was going to raise that that the thing about SARS COVID-19 that makes it unique from a MERS or even from an HIV virus is that a person can be completely asymptomatic throughout the duration of their infection.
Yes.
And someone else can die.
Yes.
And just from a public health standpoint, it creates a real communication challenge, because those asymptomatic people maybe don't really believe that they were ever sick, or if they were, they
Correct.
don't really believe that this virus is as serious as everyone says it is.
Exactly. So the
And
asymptomatic nature of these infections is it's quite vexing. It's very difficult to deal with from what you said, from the standpoint of convincing people what to do next in terms of public health. But just getting a handle on a disease pathogen that can be completely asymptomatic and transmitting to new individuals, and depending on those individuals' age or previous medical problems or things that they were born with, genetically predisposed, could have them wind up sick and dying. So that's just a very, very difficult thing to even mathematically model and get a handle on. And this is happening in earnest right now, obviously.
yeah, I would say I mean, I would ask you, it's almost like we've never seen a virus like that in our recorded times. I wonder if there's anything you could compare it to.
Not that I know of. But there is a particular thing that humans have now. It's a much longer lifespan than we tended to have a while ago, right? So the individuals on average, and I know not everyone, I mean, there are young people who are unfortunately dying of COVID, but this kind of tendency for age to scale with the problem, if you go back many years, I mean, those people weren't around. So, that itself distinguishes it from what we're dealing with right now. Now, but in the near past, yeah, it was not happening this way with other spreading diseases through epidemiology. This wasn't a concern. It didn't seem to. So I really don't have anything to compare it to. I've never heard of a wildlife disease that really has these hallmarks of the particular traits that we're dealing with here. And yet, through time, a lot of parasites and pathogens have been described, maybe somewhere in the literature, unknown to me, something similar to this had been seen, but it wasn't problematic in terms of disease, especially in humans. So therefore it was missed.
I could just totally monopolize this conversation with a dozen more COVID questions, but I know that we have a line of questions of people waiting. So I'm going to ask one more COVID question from Larry, who says, "Is COVID unique in its ability to infect a wide variety of species hosts?"
Yeah. No, a lot of viruses like rabies virus infects lots of hosts. So this is the thing that it's kind of still within virology, there's this assumed specificity of viruses to one species until you find those that are not. And I think we deal with virus problems and scrutinize them more when they're human problems. Well, somewhat same case for agricultural plants and animals, but definitely human viruses are relatively overstudied compared to other systems. So it's hard to really find generalities here, but yeah, I guess my sense of it is, as I answered earlier, that I guess I already answered. I was going to be redundant
Okay.
saying it again.
I want to go to Glenn just asked a follow-on question to the conversation we were having earlier. He says, "Is this correct? Viruses that evolve in a random environment harbor greater variation, but it's not known whether these viruses therefore become more harmful."
In this particular experiment, I think that that's a fair discussion point that comes from it. So that is one way to interpret the results. In this particular experiment, we've shown that a biological system can amass variation that's not really useful to it in its current environment, but it shows evidence that it might be useful under a particular environmental change that's not that different from the one that it is experiencing. And yeah, I think that that's a fair summary. Yeah.
Okay, I want to go to Angelo, who has a question on mRNA. I'm not sure if this is phrased right, but let's see. The
Okay.
trend you showed of 9 of 10 mRNA viruses was disturbing. Do you expect that trend line is predictive of future patterns, or are they discrete events?
Yeah, so I think what was intended to be typed was RNA, not mRNA. Messenger RNA is not RNA that's in viruses. So it's a trend that's been known for a while. I mean, I showed you data back to 2000, but if you go back to 1900, I mean, it's overwhelmingly the case that RNA viruses are what we need to worry most about as new threats. Let me say that carefully, because malaria, for example, it's a protist, right? And it's been mosquito transmitted in places where humans originally evolved, and it's still a problem. So it has literally shaped human genetics in terms of quirks like sickle cell. And why do you have certain disease states? And it's because there are certain genetics that parasite is selecting for humans to be better at withstanding it, but not really fighting it off entirely with our own defenses. Instead, we have to turn to some very strange genetic quirks to get around the problem. So I don't think it's controversial to say that RNA viruses are the thing that we should be worried about most as continued new problems. They can do this. They've shown that they can do it. They have new problems like SARS-CoV-2 that are similar enough to old problems like SARS-CoV-1. And all through time, since the 1900s, when cataloging up epidemics and pandemics of flu virus, we know that influenza virus is also problematic. It's an RNA virus. So I think it's okay. Here's what's interesting. Now, again, I said earlier, we focus so much on human disease problems that we take data from ourselves and we draw big conclusions. So I need to be careful in saying RNA virus emerging threats in humans is something we should continue to be worried about. But for example, we study some plant viruses. And if you look at most plants, they're loaded up with RNA viruses and they're not symptomatic from it. So there's something about their ability to either withstand them or shrug off their effects that RNA viruses are not uncommonly found in lots of plants. And it's not a big deal for those hosts. So what's different in their defense systems than ours is myriad things that we don't have time to go into. But I just want to be careful in terms of saying in human emerging disease, it's RNA viruses that seem to be often the drivers.
Sounds like we need to spend more time studying plant immune systems.
Maybe we do.
I'm going to go to Ruth's question. Could you say something about DNA viruses and their role in a changing environment?
Yeah, good question. So DNA viruses don't seem to have this evolutionary potential. DNA viruses tend to, not always, but tend to get inside of a host. They don't evolve very quickly, but they have larger genomes and they seem to be better capable of exploiting those genomes to change and hide out from defenses. So almost all humans are infected with at least one herpes virus, which is a large group of viruses that have DNA genomes, and they're very good at infecting, and your immune system doesn't know that it's in cells, and you kind of harbor these and you keep them. So I think that is the case in other mammals. I'm not so sure for herpes viruses outside of there. But that's an interesting question to think about, these kind of viruses that cause more chronic infections. Is there any reason to think that climate change might alter that? I'm not so sure it would. I think a lot of what I focused on today is the acute infectious viruses that are more problematic, of which RNA viruses are a great example. But I'll think more on that one. I like that question because I tend to think broadly across the virus world, and I'd like to consider the times that are here and increasingly happening to change our world and how this might affect other types of viruses than the ones I focused on today.
Well, and Cornelia asks if you think diseases like malaria and yellow fever will come back to the US with global warming, noting that they used to be prevalent in the Southeast, but also in the wetter parts of the Northeast.
Yeah, good question. I mean, not necessarily, because it depends on the vector species of mosquito for these various pathogens. And that's actually something beyond my expertise. There's a wide variety of species of mosquitoes. Some of them are sweeping the globe and increasing their range size. Others are less capable of doing that. So again, we're so laser focused on the vector arthropods that are problematic for us. Yeah, and again, for our agricultural animals, such as here in the Northeast, there are definitely some mosquitoes that transmit viruses that can be deadly to horses and would be quite concerning with outbreaks like Eastern equine encephalitis virus. So I think that's a great question. And here, I guess I could recall some discussions I've had, unfortunately, with mosquito ecologists, that it's not, it's like through time, there's a decreasing number of experts in mosquito ecology for reasons that I won't get into. So it's a little worrisome that for some things within public health and disease concern, it's not as if everybody's being trained in everything. There's always things that are more exciting fields for people to run to, and those that maybe are less populated. And it turns out that the ability for somebody to sit down and distinguish between one mosquito species and another one based on its visible traits is not that common anymore. So that was a long-winded way of saying that various mosquitoes could be expanding their range and others wouldn't necessarily do that. And I kind of gave it as a general example, again, driven by our concerns in human disease. So that's a good question. If we can get enough people who are into mosquito ecology and range expansion and distinguishing between these species and sampling a lot from environments to do the surveillance, then we could get a handle on it.
Okay, so we need more Yale PhDs in mosquito ecology.
There you go. I like that.
Gary wants to know what adaptation or evolutionary factors allow viruses to jump species.
That's a lot of the research that we do in my lab that I didn't really have time to talk about. I'll go quickly to my take on this, is that organisms that exploit lots of resources, we call them generalists. They do have different traits than organisms that are very keyed in on only one or few resources, which we call them specialists. So yeah, actually, let me, people think about bats a lot these days. I'll give you an example. Fruit bats, which are daytime foragers. They fly around, they roost in trees, they eat lots of different fruit. Vampire bats only eat mammalian blood, right? So they have a very, very limited diet comparatively, but they are also bats. So again, wait, the main point of the question was which traits was it? I forgot the main point. Oh, you're muted. Sorry.
Basically, what allows a virus to jump species?
Okay.
Species, what adaptation?
Okay, so sorry, I forgot that. Lost my train of thought. Talked about bats too much. So my take on this is that immunology matters. So host defenses occur, and let's take mammals, which have immune systems, both innate and adaptive. So you have these defenses that you're just born with that are innate defenses, as well as those that could be molded through time. We call that adaptive immunity. So my expectation, based on observations and things I've read in the literature, is that viruses that have evolved through time to exploit multiple host species are quite good at navigating host defenses to evade them or avoid them. And this could predispose them to jump into another mammal that is a relative, because all mammals have some recent common ancestry. So even though you have, very different mammals like armadillos and rhinos and humans, there are some commonalities in their immune system. So is it really the case, perhaps, that certain viruses evolved to generalize by navigating host defenses successfully? And you can think of that as like a network that has some nodes of commonality from host species to host species to host species. Even a virus with a handful of genes, if it's keying in on that node in the network, that it has to overcome it in order to replicate in the host, then maybe that's the only thing that matters for it. And it could very easily move between host species. So that was one particular example in addressing this question of the navigating and overcoming immune defenses could be something that if a virus is groomed to do this across lots of hosts, what's the big deal about jumping into one more like humans? So that's what I think we should be looking for, are viruses that are known to already exist in lots of species, and do experiments and on them to see whether what I said is really true generally. Is it something about generalist viruses that are better predisposed to jump. It seems to be the case because a lot of these viruses that we're dealing with now, including SARS-CoV-2, you hear reservoir species talked about all the time. And nobody is claiming that they even know the extent of reservoir species. And I have to say, I didn't read the article yet, but I did hear the kind of bad news that I think it was a herd of deer in Iowa were shown to be loaded up with SARS coronavirus 2. It's like, oh boy, I could have seen that coming, because it's a virus that's already known to jump from other reservoirs into humans. Why would it not be jumping into other organisms as well? So that's also part of our research, is to look at particular mammalian species and try and do the hard work of figuring out how vulnerable are they or not to the entry and the infection of SARS-CoV-2 to make those animals reservoirs.
That is the topic of another presentation.
Uh-oh, I better not say any more.
Invite you back to give.
Sorry. Tune in, folks, next time, because I'm sure my armchair account of that was nothing like what you're going to hear.
No, that was definitely the thesis of a whole entire additional presentation.
Wow. Awesome. Okay.
I want to go to, there's a lot of really interesting questions. So if you guys will hang with us, I'm going to try to get to all of your questions. Christina wants to know, are you concerned that melting glaciers will release ancient viruses which could be more virulent? And I think you sort of speak a little bit about that when you're talking about, these generalist viruses that have developed the ability to jump. So theoretically, a virus frozen in a glacier wouldn't really know how to get to a human, but I wonder what your take is on that.
Yeah, so I've seen that sci-fi movie. I've read that book. And the problem is I'm a sci-fi fan. So I'm like, well, it's plausible that something, I think it's actually more, okay, so two things I'll say to this. It's pretty plausible for an old bacterium that is quite good at doing something called sporulation. That's sort of, a way that it changes to ride out very difficult environmental conditions. We know that some bacteria can thrive for hundreds, if not thousands of years doing that. So if you have melting permafrost and suddenly you have some bacteria that are awaking, then maybe they would still be capable of causing infection. But as you mentioned, there would have to be these hosts around, and there would have to be some chains of transmission in order for us to even know what's happening and to be a problem. Now, viruses, you don't have quite this ability that we know of for viruses to persist for very, very long periods of time, just outside of cells. Some can, and that's actually how we store some of them in our freezers here, is we just put them in liquid, no other additives except basically liquid, and we stick them in a freezer, they freeze solid, and we thaw them and we use them in our experiments. So that's plausible as well. I don't know how common that would be encountered in nature. The thing I would worry about more would be some humans are buried in the permafrost. If they happen to have died locally in those areas and were buried, and they may not be very deep graves, they could be shallow graves. So it is definitely plausible that if there's any remains there, it's very good. You can get very good mummification in permafrost by simply just the dryness and the coldness. So if you thaw that, then yeah, that's why you find, baby mammoths in permafrost, is that they're mummified. So I don't want to make any claims for evidence that I have zero to back up the statement, but I think it's an interesting question and plausible enough to think, as the ice caps melt, as places that are historically cold become warmer, and if you have humans that are buried there that have died for some diseases that we actually know of, should we be worried about those diseases emerging again from them? And I think the obvious example would be smallpox, which is caused by variola virus. When smallpox was a big deal, for which it has been a very big deal until very recently, when we did a global vaccine campaign to get rid of it, there were lots of people dying in places where they were buried in shallow graves in then cold environments that are no longer so cold. So I don't know if the right chain of events would happen for that to be problematic.
Very disturbing indeed.
Sorry.
Um, I want to go to Lena's question. Lena asks, if viruses are adapting so well to higher temperatures, what do we do to counter them?
Well, good question, Lena.
I realize that's not the side of things that you work on, but I
So
thought
that's okay.
you might
No,
want
but
to throw
no,
out
it's oh, I'm
some
happy to. Hey,
theory.
look, I'll answer any question if somebody gives me time. So the ability for, say, mRNA vaccine platforms and other things that have been developed recently, we have different types and a greater variety of creating vaccines. And to be clear, that's a new technology that is very likely to work very well for some targets, but not others. And that's the whole reason that we have lots of different ways that we make vaccines. It depends on what the target is, to how you formulate it and create it, about whether or not it's going to be useful. So I guess as a scientist, I have to say something on the side of science, is that we make breakthroughs all the time. And therefore, I always have some optimism about challenges of the future and the ability of technology and science and ingenuity to get us through them. So the funding has to be there. I could say all sorts of things about the underlying assumptions for that to work. But I think the challenges of any biodiversity problem, whether it's infectious disease increases or species losses, as the world warms and climate change becomes more of a problem, I don't think there are any hard and fast solutions. And the best solution would be to reverse the problem, right? I mean, and then hope that it kind of reverses itself and goes away, which is a bit naive. Now that we got ourselves into this, it's not that easy, as people have reminded us who actually work on this stuff, to say, oh, just, stop and we'll go back. And oh, that may take a very long time. So I think it's a great question. And I suppose in the end, I'm rambling a bit, but I would admit that more research, it's kind of that slide where I showed, if you read reviews on climate change and what are our expectations for infectious disease problems of the future, all of them admit that we need more information. We need better tools or better insight into our existing tools to really dig into these problems and get more information that might help us in the future. So I guess I'll reiterate that.
Okay, I want to take another. We have just a couple of minutes left, but there are two kind of interesting questions that take a different angle on this altogether. So I'm going to combine Stephanie R's question with Stephanie says, when you look at lower latitudes on the globe, there's greater biodiversity at lower latitudes. So if you think about the warming of more northern latitudes, then you might expect to see an increase in biodiversity and species, new species developing. And what does that do to viruses? What does that do to the prevalence of viruses? And I want to combine that with Natalie's question, because I think it's an interesting combo. Is there any way to model how possible predators of infectious diseases might evolve or compete with regard to the infectious microbes themselves? So I'm sort of asking a hybrid question: is, as the climate changes, species also change. So we're worried about the viruses, but what about the predators to the viruses and how they might evolve or arise?
Yeah, interesting question. So the first part of it was, in a warming world, yes, around the equator, the idea is that the more northern latitudes are also going to be warmer. And if it is merely the fact that higher temperature gives you more biodiversity, of which I'm not so certain, there's a lot of nuance to that, right? I mean, there's different rain patterns. I mean, you're talking about the continents, and there's a lot of difference at the equator versus the higher latitudes. And it's a bit like the mountainside example I gave, that if things get warmer in the lower northern latitudes and the southern latitudes, right, as you move away from the equator, it's also going to get warmer in the equator. So the problem is, would you turn places that are kind of more thriving and lush communities and tropical forest and all that, and they become more desert-like? I mean, that's a very extreme. That's going all the way to one end of the spectrum. But essentially, yeah, you can get places that get too warm to sustain their current species. And if those species, as I said, if they can't move, if they can't disperse, or if humans are already in the way so that their natural habitats are not available as they try and move to other latitudes, then they are probably going to go extinct. Now, the question about predators of viruses is an intriguing one, because what is a predator of a virus? I actually don't know of any. It's kind of your immune system cells are predators of viruses, because if you're infected, you recruit certain cells that are good at traveling around your body and finding them, unless the viruses are good at hanging out, like herpes viruses are, and are never found and detected. So if the question means, so I'm going to go back to my science fiction hat and just make things up, right? So nanotechnology, if any of our listeners know what that is, it's basically teeny tiny robots, for example, and things that humans can manufacture. So if we could use those, which are, not that different in scale from a virus, as something that could also, and this is definitely science fiction, right?
It's not, it's actually not that far off, because I've seen writing about this at MIT, that they are working on these sort of nanopathogens that they can introduce into the human bloodstream, right? So you're saying it's sci-fi, but it is happening.
It is. This is true, Lauren. It's happening. How soon would it be approved for human therapy? That's kind of where I backed away and said it's sci-fi. I mean, I work on viruses that are trying to hunt around your body to kill bacteria that antibiotics won't kill. And it's actually working very well. We treat emergency patient cases and save their lives, and we're doing clinical trials, but there still is no such virus approved in the USA by the FDA as a drug, right? So we're still not at that point. And I like what you said there about at MIT and other places. Indeed, that technology is being examined. Here at Yale, it's being examined. So it's interesting. Can we sort of do microbial warfare, right? To do surveillance and remove and attack some foreign invading disease pathogen that is a microbe. I'm trying to do it with naturally occurring microbes that is the enemy of my enemy is my friend, right? But as we design things that could serve that purpose, it's, I guess, currently science fiction if it's not approved, at least that's what I would define it, but it's not that far off potentially to be going into clinical trials itself. And for all I know, that's already happening. I mean, it's really exciting. However, I should also say that I have written government reports and other things about if you have technology and you can do things, you need to be careful with that technology so that it doesn't get out of your control and it's really doing what you intended. So I am into enough of bioethics to study that problem. And there are many exciting ideas out there for which we also know that there's a lot of toxicity involved in them, right? And so we have to be careful that not every technological advance goes so smoothly that it really leads to something that is a breakthrough that is used to solve human problems. Some of that is fast and some of it is quite slow.
Well, there is another thesis for another presentation. So we've got you on the hook for 2 more after this.
That's great.
Thank you very much, Professor Paul Turner. I want to thank also Glenn and Gary, who posted stories about the permafrost releasing anthrax in Russia. And so there you have it. Your theories have already been proven. This is what I love about our Yale alumni events. There's someone in the audience who knows the answer to any question that we could ask. So thank all of you for your participation. This has been quite an interesting discussion. We've gone over a little bit. I do want to give just 1 more minute for you, Professor Turner, to talk about, I believe there was a certificate program that we wanted to make sure we mentioned to everyone if they're interested.
Oh boy, and I'm supposed to have the information on that?
I think it was.
Oh boy. Is this a global health certificate program?
I think that was it. Yeah.
Oh boy.
Just to say that
I think it exists. Well, maybe I can back away from that question and go do an interrupt. Is that there's a lot of global health interest at Yale. I mean, this is literally where I was right before I joined you all, is that we had a group of faculty presenting publicly and to other faculty about all the many ways that we feel like global health problems could be, creatively solved or addressed. And we describe the many ways that the Yale faculty were trying to do that. So, yeah.
Well, there we go. Thank you for that. Thank you for joining us. We'll look forward to the next one.
Thank you.
I thank all of you for participating, and we'll see you hopefully on Monday for our final climate change conversation. Bye-bye.